We'll now look at the antiviral drugs. We'll look at those that target a unique enzyme DNA polymerase in varicella zoster virus and herpes simplex virus. We'll look at antivirals that target influenza A and B viruses, and we'll look at antiretrovirals in the treatment of human immunodeficiency virus. We'll also explain why there is such a small number of viruses for which there are antiviral drugs. With viral infections, it's a lot quicker than a bacterial infection. The virus replicates very quickly in the host cells. It's very quick then to show clinical symptoms, but often the virus has already been shed and has actually been infectious for a while before those symptoms appear and before the viral content has reached its peak. Viruses have very few unique targets. It's this idea that they are obligate intracellular parasites. They must take over the human cells to get access to all of the machinery needed to manufacture their own genome. If it's a double-stranded DNA virus, then they will use the same machinery as the human cell to replicate. Same with single-stranded RNA viruses. They will use ribosomes of the human, the mammalian ribosomes, to make the viral proteins and to assemble viral particles, and to use the ribosomes to then manufacture more genomes, more single-stranded RNA to go into those particles. So the capsid is the protein surrounding that genome, surrounding the DNA. So there are very, very few unique targets, meaning that there will be very few antiviral drugs available. Any drugs that you do use could have the effect of disturbing the host cells, could be very toxic to the host, and so antivirals not terribly common. Antivirals are given as chemoprophylaxis to prevent the virus from replicating. Once a cell has already produced viral particles, there are very few antivirals that actually will then kill those already assembled viral particles. They can only prevent them from then infecting other cells. So to look at the way that viruses replicate, obligately inside the cell of the host. They have to attach via various proteins. The genome is injected into the cell. It takes over all of the enzymes of the cell and uses it to make viral proteins, viral DNA, and then there is self-assembly into particles which then kill the cell as they are released. These particles will then attach to an uninfected cell and the infectious cycle will spiral. There are certain antivirals that target DNA polymerase and inhibit this particular enzyme. So we said that this was for the prophylactic treatment of herpes simplex virus and in the treatment of varicella zoster virus, which causes shingles in adults and chickenpox in children. But it's for the shingles that the drug acyclovir is indicated. This is sold as the proprietary brand Zovirax, but there's also famciclovir and others. The mechanism of action: the drug is taken into the host cell. It's converted to acyclo-GTP. This is the active form of the drug, a metabolite. This acyclo-GTP inhibits DNA polymerase. The acyclo-GTP becomes incorporated into the viral DNA, where it terminates the synthesis of more viral DNA particles. Adverse effects of the DNA polymerase inhibitors such as acyclovir: gastric distress, headache, dizziness, nausea, diarrhea and vomiting. Acyclovir and other DNA polymerase inhibitors will interact with probenecid. There'll be increased levels of acyclovir and the adverse neurological effects will be seen. Warnings and contraindications: use with caution in people with neurological impairment, kidney function impairment, or dehydration. Neuraminidase inhibitors are used to prevent influenza A and B. So once again, a prophylaxis. They're for adults and children over the age of 1. These drugs, such as oseltamivir, target neuraminidase, an essential enzyme for the replication of strains of the influenza virus, particularly A and B. This enzyme is needed to facilitate release of the virus from infected cells, thus enabling it to then transmit and infect other cells of other hosts or other cells in that body. So this class of drug, by inhibiting the action of the enzyme, actually prevent the release of more viral particles. Neuraminidase inhibitors, adverse effects. It does not induce metabolizing enzymes, so drug interactions not as likely. Adverse reactions include the gastrointestinal effects and some CNS effects, dizziness and vertigo. It's used with caution in people with an underlying respiratory or cardiac condition and a complicated influenza where it has already progressed to a pneumonia. They should be taken within 48 hours of the onset of symptoms and taken with food to reduce any stomach discomfort. There are 4 drug classes that are used to target HIV. These are the antiretrovirals. The first one that was developed was zidovudine, or AZT. Zidovudine is a nucleoside reverse transcriptase inhibitor. So the way it works is by targeting a unique enzyme in the HIV. And this enzyme is the enzyme that's needed to make DNA from RNA, because this is an RNA containing virus. If you can prevent that RNA from producing DNA, you can suppress the replication of the HIV. Once again, it won't have any effect on T cells or other antigen-presenting cells that already contain HIV, but it will prevent any replication in other cells. So the indications for nucleoside reverse transcriptase inhibitors like zidovudine, HIV and AIDS, where the T4 cell count, the lymphocytes that contain that CD4 antigen, is less than 500 microliters of blood or less. These drugs can also be used to prevent vertical transmission of HIV during pregnancy to the neonate, and this would be in the 2nd and 3rd trimester, contraindicated in the 1st trimester. Adverse effects: bone marrow suppression, anemia and headache. The bone marrow suppression being quite serious in people who already are immunocompromised. Then there are those that have been developed that are non-nucleoside reverse transcriptase inhibitors, such as efavirenz. So here it's blocking RNA-dependent and DNA-dependent DNA polymerases. It binds to retroviral reverse transcriptase near a catalytic site and denatures it. So these drugs, such as efavirenz, are indicated in HIV infection and preventing that mother-to-baby transmission. Adverse effects: gastrointestinal and fever, contraindicated and used with caution in people with impaired liver or kidney function, a rapid emergence of a viral infection, and they will interact with the oral contraceptive pill, decreasing its effectiveness, and there is increased metabolism of methadone with these non-nucleoside reverse transcriptase inhibitors such as efavirenz. Another class of antivirals against the retrovirus, antiretrovirals, the protease inhibitors. These drugs, such as sequinavir, act by blocking the viral precursor polypeptide, blocking the maturation of the virus. So those polypeptides have to mature by being processed, and this process is completely blocked. Adverse effects of the protease inhibitors: headache, diarrhea, nausea, vomiting, so the CNS and gastrointestinal symptoms, but there will also be elevated liver enzymes showing that there is some damage to hepatocytes. There will be hyperglycemia, hypertriglyceridemia, and hypercholesterolemia. These protease inhibitors, such as saquinavir, do inhibit a particular isoform of cytochrome P450 enzymes. This will lead to increased levels of other drugs. Then there are those that are viral fusion blockers, such as enfuvirtide and tenofovir. The mechanism of action: there is a synthetic 36 amino acid peptide that binds to the glycoprotein 41 subunit of the viral envelope and blocks viral fusion with the CD4 receptor on the host cell, and this will prevent conformational changes that are required to allow fusion of that viral particle with its host cell. T4 cells, as we said, very rich in that CD4 receptor and also expressed on antigen-presenting cells. The viral fusion blockers are indicated for, once again, HIV infection, but to HIV-1. This is used where other treatments have failed. Adverse effects include injection at the site. There is often reactions, insomnia, depression, respiratory symptoms, loss of weight and appetite. This is in people who are already quite immunocompromised and makes their symptoms a lot worse. In the treatment of HIV, it is seen that no one therapy works. Zidovudine very quickly resistant strains of viruses were found to it, so to prevent any resistance appearing in any of these new chemicals that were produced, these combination treatments were developed, and one of the best is the highly active antiretroviral therapy, the HART procedure, where you give 2 nucleoside reverse transcriptase inhibitors, so 2 of those with one other, either a non-nucleoside transcriptase inhibitor, or you would give 1 or perhaps 2 protease inhibitors. Inhibitors of HIV fusion, such as enfuvirtide and tenofovir, are used where resistance has developed to those other drugs. In HIV treatment, post-exposure prophylaxis might be indicated. Individuals are given a course of drugs to prevent initial viral replication. So combinations such as Viracept and Combivir. Combivir is lamivudine and zidovudine, and Viracept is nalfinavir. In summary then, the sites to target HIV, some rather unique sites. The nucleoside reverse transcriptase inhibitors, abacavir, didanosine, emtricitabine, lamivudine, stavuvudine, zalcitabine, zidovudine, the very first one. The non-nucleoside reverse transcriptase inhibitors, delavudine, efavirenz, nevirapine. There are others as well that will also work on the genome of the HIV, such as tenofovir. Then there are those antivirals that will target the proteases that are needed to mature the developing polypeptides, the protease inhibitors. Aprenavir altazanavir, fosamprenavir, indinavir, lopinavir or ritonavir, nelfinavir, ritonavir, and the one that we looked at, which has also effects with grapefruit juice, was saquinavir. Then there were those that will target fusion of the viral particle with the cell surface receptors. So we saw that there has to be binding between the viral particle and a particular chemical, CD4, on a host cell such as a T cell or an antigen presenting cell. And a drug that would block at this site would be enfuviratide. So if that is not blocked and the viral particle attaches to the receptor, there is uncoating and entry into the cell. The RNA is eventually released. The reverse transcriptase was in that capsid-contained package, and that reverse transcriptase is then used to take that RNA genome and make viral DNA. The DNA enters the nucleus, it is integrated into the DNA of the host, forming a provirus, part of the host genome, and as such it is replicated and it is transcribed along with the normal host DNA. Messenger RNA is made, it takes over the cellular machinery of the ribosomes and uses the host's ribosomes to then manufacture more viral proteins. Replication of the DNA to make new genomes, which are then excised. They are not part of the host DNA, they are used then for assembly of new viral particles. The immature virion is not quite infective yet. It first has to leave that host cell, and in leaving it then gains a lipoprotein envelope where the lipid component came from the host, and some of those protein particles are assembled HIV proteins.